Okay, our next speaker is a graduate student at UC Davis. His name is Kai Battenberg, and he's going to talk about the association of coyote ceanothus with frankia, the nitrogen-fixing microbial symbiont.
So first, I'd like to thank everyone for coming to this talk. The title of my talk, as you mentioned, was Can They Live Without Each Other? The Strength of the Association Between Ceanothus ferrisiae, an endangered California native plant, and frankia, the nitrogen-fixing microbial symbiont. For those, the very few of those who missed the first part of Janelle's talk, Ceanothus ferrisiae is a perennial shrub that belongs to the family Rhamnaceae. It's endemic to California. It's endemic to serpentine soil. It's endangered with only 3 populations left in Morgan Hill, south of San Jose, and Anderson Reservoir. So there are 3 populations left, again, as Janelle was talking about it. The largest population is the Anderson Reservoir population, indicated as a combination of AR and WA in the map. The youngest population we did not indicate on the map because we did not use in our specific study. The third site, the Kirby population, is indicated as K. You'll also see that at the bottom right corner of the map is the Anderson Dam that is planned for retrofitting, and the Anderson Reservoir population is expected to sustain significant damage. So the plan is to transfer Ceanothus ferrisiae to CC site indicated on the map to establish a new population. There are 2 things that's unique about Ceanothus ferrisiae. One is that they are serpentine endemic. Now, serpentine soil is characterized by their low calcium to magnesium ratio, usually defined as less than 1, which makes it toxic for most plants. However, once you get adapted to such an environment, because other plants have a hard time living in such a harsh environment, it often serves as a refugia for many native plants. Now, in our sites, the CC site, K site, and AR site, indicated as red shade, are all serpentine soil, and then one corner, the WA site, that's indicated as blue, was determined non-serpentine. The other thing that's unique about Ceanothus is that they establish root nodule symbiosis. So the host plant provides photosynthate to the nitrogen-fixing microbial microbe frankia, and in return, frankia fixes nitrogen from air and provides nitrogen in the form of ammonium to the host plant. And this all occurs in this specialized organ called the root nodules underground. One thing that I would like to mention is that, that this is similar to, but is unique from, the legume and rhizobial symbiosis. They're different partners. One more thing is Ceanothus in general is known to establish this association. However, Ceanothus ferrisiae in specific has not been studied up until this point. One more thing that I'd like to clarify is that when I say frankia, I am in fact talking about one specific subgroup of frankia known as the cluster 2 frankia. This is the only group of frankia that is able to associate with Ceanothus. So the 2 questions that we were asked to answer for this project was 1, does Ceanothus ferrisiae fix nitrogen in native serpentine soil? And 2, is frankia present in CC site soil? Through answering these questions, our goal was to answer, does native soil of Ceanothus ferrisiae need to be transferred with the plant to CC site or not? So one thing was that we took soil samples to test if a given soil is present or absent of cluster 2 frankia. We were able to collect a total of 18 soil samples from points indicated on the map. And along doing this, we were also able to partially excavate 3 plants, 1 from WA site and 2 from AR site. From each soil collected, we performed a DNA extraction, and from each DNA extract, we performed a polymerase chain reaction, that's PCR, to detect cluster 2 frankia specific DNA sequence. If present, then we assume that that given soil was present for cluster 2 frankia. We also collected leaf samples to test if a given plant is fixing nitrogen or not. Of course, the best way to show nitrogen fixation is to excavate the plants and find nodules, and that's the best way. However, we have to keep in mind that Ceanothus ferrisiae is an endangered plant, so we needed a rather non-invasive method. So we collected over a total of 100 leaf samples from 4 species, including Ceanothus ferrisiae, our putative nitrogen fixer, and Artemisia californica, that's a known non-nitrogen fixer that's abundant on the site. So, as I mentioned, we needed a non-invasive method to test for nitrogen fixation. We decided to use delta-15N stable isotope analysis. Nitrogen has 2 stable isotopes, 14N and 15N. A nitrogen-fixing plant, such as most ceanothus, is getting their nitrogen from frankia, and frankia is getting their nitrogen from air. On the other hand, a non-nitrogen fixer is getting their nitrogen from soil solution. When you compare air to soil solution is relatively enriched for 15N. So, if you look at the nitrogen composition of a leaf sample taken from a nitrogen fixer and compare that to a non-nitrogen fixer, it will be enriched relatively for 14N. So, delta-15N stable isotope analysis is able to detect this difference. So based on these methods, the first thing we found was that Ceanothus ferrisiae is in fact able to form root nodules. The one plant that we were able to partially excavate in WA site was forming nodules. Unfortunately, this was a non-serpentine soil, so we were still not able to say that they were fixing nitrogen in native serpentine soil. However, we were able to show the capability of nodulation. Then, out of the 10 soil samples that we collected either from K site, AR site, or WA site, all came back positive for frankia. To make sure that this was in fact a cluster 2 frankia, I also performed phylogenetic analyses to compare with other known cluster 2 frankia. And you see that all the samples that are shaded in gray were clustering together with the other groups that are known for cluster 2 frankia. So, in essence, we found that Ceanothus compatible frankia is present in native serpentine soil. So based on the delta-15N analysis, we found that when we compare Ceanothus ferrisiae to Artemisia californica, which was a control, we saw that Ceanothus ferrisiae has a lower delta-15N, which means that it is more enriched for 14N. So that is consistent with nitrogen fixation. One more thing I'd like to point out is the average delta-15N of Ceanothus ferrisiae samples collected from AR site and K site combined. These are where it's serpentine soil. And compare that to the pink bar, that is the average delta-15N taken from Ceanothus ferrisiae that was growing in WA site, where nodules were found. We did not find a significant difference between the two. Now that is what you would expect if the two groups of plants are in fact using the same source of nitrogen. So again, consistent with nitrogen fixation. So all those 3 points combined, Ceanothus ferrisiae is able to form root nodules. Ceanothus compatible frankia is present in the native serpentine soil. And Ceanothus in serpentine soil and WA site, where the nodules were found, are using the same source of nitrogen. All those combined, we determined that Ceanothus ferrisiae is fixing nitrogen in native serpentine soil. Also, of the 8 soil samples that we collected from CC site, every single one of them came back negative for frankia. So, which meant, to conclude, upon transferring Ceanothus ferrisiae to CC site, native soil should also be transferred. So, this is the last slide that is related to the project of transferring Ceanothus ferrisiae to CC site. However, while I was doing this, it got me wondering, exactly how strong is the association between cluster 2 frankia and the host plants? Because from experience I know that you can grow Ceanothus in the greenhouse without cluster 2 frankia so long as you provide them with nitrogen. On the other hand, if you look at from the perspective of cluster 2 frankia, there are some mixed results. Of all the frankia genomes sequenced, the smallest one is coming from a cluster 2 frankia. Now, in small genome sizes, often correlated with parasitism. It doesn't prove it, but it's consistent with it. And unlike other strains of frankia, cluster 2 frankia is so far unculturable, indicating that whatever environment that is that the host plant is providing is unique and difficult to replicate. Finally, as we saw in CC site, it has not been found in soil devoid of host plants. So all of these suggest that cluster 2 frankia in fact does need the host plant to be in the vicinity. On the other hand, if we look at the biology of root nodule symbiosis, it's weird because if nodules are forming in the soil, that means that the frankia has to be in the soil by the time the root reaches that particular site, suggesting that they are in fact able to survive in soil that's devoid of host plants at that point. This somewhat weird, interesting interaction got me interested enough to replicate this experiment at a different site, further up north in California in McLaughlin Natural Reserve. Here I took a total of 55 soil samples, and previously in Anderson Reservoir, all sites except for WA site was serpentine soil. So in this case, I wanted to make sure and find 2 sites that were not serpentine soil to make sure that whatever observation I make is not restricted to a one given site or one given soil type. So 2 sites were serpentine soil, 2 sites were not. Of each pair, 1 site was present with the host plant, the other site was absent with host plants. So when we zoom into the map, this is still in progress, so you see dots with 3 colors. Each dots indicate the place where I took my soil sample. The red dots are the ones that I have not processed yet. The black dots are the samples that came back positive for frankia. The white dots are the points that came back negative for frankia. So 2 things that I found was all samples that were taken from host plant positive sites also came back positive for frankia. All samples that came back negative were taken from sites that were absent of host plants. And this pattern was not different in serpentine soil or non-serpentine soil, suggesting that it's not restricted to a particular soil type. Now, unfortunately, I didn't see a perfect correlation that I saw as in CC site, but one thing that I'd like to mention is that here, whereas in Anderson Reservoir, most places were lacking a host plant except for AR site, WA site, or K site. Here, most of the part that you see in the map is in fact full of ceanothus. These 2 sites that were absent of any host plants are rather an exception. So the samples that came back positive of frankia on sites that are absent of host plants may be due to the fact that there's some soil that's coming in from the surrounding environment, or that the host plants are not currently there but used to be there, and that's what's remaining. So it seems as though that from what we observed, cluster 2 frankia can survive without the host plant, however not indefinitely. Of course, I'm hoping to find out more once I process the remaining data. The take-home message here is, upon transferring any nitrogen-fixing plant that hosts cluster 2 frankia, not necessarily just ceanothus ferrisiae, you should at least consider transferring the native soil as well. Finally, I would like to thank Janelle for putting together the project, and my advisor, Dr. Allison Berry, and the undergrad that did most of my soil sample analysis, Jenna, and Mujing for doing the initial sampling in Anderson Reservoir, and of course, California Native Plant Society for giving me the funding to do the experiments in McLaughlin Natural Reserve.